We are in a field that is booming now with a new discovery every weeks or months, with new knowledge, with new model system, with new technology and so on. And that's super exciting because we are really the thing that we are at the forefront of this new wave of discovery. And it's very exciting to see that we can really have an impact, and all the projects we develop are part of this massive progress.
Ask Different, the podcast by the Einstein Foundation.
Hello, my name is Doris Helpold. Welcome to this episode of Ask Different. If you're into Greek mythology, you might know that Prometheus was punished in a pretty harsh way by the god Zeus for bringing fire to humankind. Tied to a mountain, he was supposedly being attacked every day by an eagle who fed on his liver, which then always grew back by the next day. A tiny part of that myth is, in fact, medical reality. The human liver can indeed grow back when a part of it has been removed. A lot of patients with acute or chronic liver failure rely on that very fact as they wait for or are lucky enough to receive a liver transplant, because in some cases a diseased or damaged liver can actually be replaced by a portion of a healthy liver from another person. However, some people need whole liver transplants, and generally the number of people waiting for liver donations is far higher than the actual number of available transplant livers. The Central Institute for Public Health in Germany, or Bundesinstitut für öffentliche Gesundheit, for example, regularly publishes statistics for organ donations. And according to their website, in 2022, there were about 750 liver transplants in Germany. And at the same time, there were some 840 people on the waiting list for a liver donation. So clearly, there is a need for solutions for patients who might otherwise not survive, or at least be severely impacted in their quality of life. This is exactly where the research of today's guest on the podcast could potentially play a vital role in improving the situation for such patients. Ludovic Vallier and his team at the Berlin Institute of Health are working, among other things, on something called organoids grown from stem cells, which might hopefully lead to viable treatments and therapies in the near future. Thank you for being with us today, and a very warm welcome to you, Ludovic Vallier.
Thank you. Thank you for this nice introduction, and thank you also for giving me the opportunity to present the work of my group today on the podcast. And hello to everybody.
You started working in Berlin 3 years ago following an invitation by the Berlin Institute of Health at the Charité for an Einstein strategic professorship, specifically to research stem cell use in regenerative therapies. And most recently, before that, you were based at the University of Cambridge, doing similar research at the Cambridge Stem Cell Institute. So I think it's more than fair to call you an expert in that field. Also, excitingly, at the end of last year, you've been awarded an ERC advanced grant by the European Research Council, which comes with 2.5 million euros over a 5-year period. Congratulations. As far as I understand, it's been the third time you got this award, which is, I guess, a rare privilege and shows how much interest and importance is placed in the kind of research work you do. What exactly are you going to use these funds for over the next few years?
So yes, as you mentioned, we're looking at the liver and how the liver can regenerate. Like the Prometheus lesion, now we know that the liver can repair itself very efficiently, especially in acute injury. If you cut the liver, you can cut 2/3 of the liver and it will regrow in a few weeks in human. That's actually the frontline treatment for liver cancer, that you resect part of the liver that contains tumor and the liver of the patient recover. So we know it works in acute injury, but it's more complicated in chronic liver disease that represent by far the highest number of patients, and especially diseases that are affecting the liver over a very long period of time. And in this case, we don't know exactly what are the mechanisms of regeneration and if the liver can really repair itself. And so the goal of this grant with the ERC is to understand those mechanisms of regeneration in chronic disease, and also to understand those mechanisms to promote those mechanisms of regeneration, so to promote the repair of the tissue, and why avoiding any problem, because we know also that this regenerative mechanism may be also associated with cancer. So we want to promote regeneration and block tumorigenesis at the same time. And that's really trying to understand the mechanisms that are beyond this fine balance that we're going to use this funding for, and then hopefully develop new treatment that will allow us to promote tissue repair and block liver cancer.
Can you give us some examples, perhaps, of chronic liver disease that you might possibly be addressing at some point in the future?
So the main chronic liver disease we're looking at is called non-alcoholic fatty liver disease, or now also MASLD. It's basically a disease which is quite common. In fact, no, it's accumulation of fat or lipids in the liver. It's often associated with overweight, obesity, or diabetes. And in this case, as the liver is the main organ that processes lipids, when you have too much lipids, basically the liver starts to accumulate too much lipid. And 30-40% of the population, at least in Europe and in the US, will have some fatty liver. So it's not a problem, but in some cases, this accumulation of fat in the liver becomes toxic. That's create an injury, which takes a lot of time to develop, but progressively, go to cirrhosis, which means basically liver failure. And we are focusing especially on this disease because there is a booming number of patients that are suffering from NASH, or non-alcoholic fatty liver disease. And we're really interested to understand what are the mechanisms of regeneration in the context of the disease, and how we can basically stop the progression of the disease and repair the tissue.
I find it amazing that liver cells or liver tissue has these properties of regeneration. Is it possible to explain how that works, or is that exactly the kind of research that you also do, trying to find out how that happens?
So that's exactly the question we want to ask. Because, it's funny, as you said, it's known for a long time that the liver can regenerate. And the Prometheus legend is a legend, but it's known at least from the 1800s that the liver can regenerate. And so far, we don't really know in detail the mechanism that are involved in this regeneration, especially how the cells can reproliferate and regrow the organ, like how the organ regrow. That's really not fully understood. And also how the organ can regrow the same size and the same level of function. And that's really fascinating because, in fact, the liver is the only organ capable to do that. Of course, if you cut 2/3 of your brain, your gut, your kidney, it will not regrow. It's only the liver that can do that. It's been almost 2 centuries now that we know the liver have this amazing property, but we still don't know the mechanism. And that may be an important message here to give to the audience, that while the liver for a long time have suffered from a negative kind of publicity, because one of the leading cause of liver disease is alcohol abuse. And as it's self-infected, for a long time, the liver was seen as bad organs, or not that interesting organs, which means that for a very long period of time, the research on liver was underfunded. And that's really created a gap of knowledge, understanding of disease mechanism, regeneration, and basic biology of the organ. Of course, as I mentioned, it's changing very quickly now because of the emergence of fatty and non-alcoholic fatty liver disease, which is now the predominant cause of hospitalization for liver disease. But for many years, in fact, research has been limited in the liver because of this dogma around alcohol abuse. That's a good example of where you can see that public funding is really important to support research. And the gap of treatment we have now on liver disease is coming exactly because of this lack of funding for many years.
That's very interesting, actually, how much the image of a disease can impact the level of funding. But that's not a question we want to address in detail today. As far as I understand, how you go about finding out more about the regenerative properties of liver tissue is that you use actual liver tissue, but then try to grow it on in a lab dish to create like mini livers. Is that fair to say?
Yeah, so the philosophy in the lab is really to work on human patients. So we generate data from human patients. We access often liver biopsy, which are basically pieces of liver that are taken from patients for diagnostic purposes. And we use part of those biopsy to study cell behavior and basically to study the disease. And at the same time, we also derive from the same biopsy what we call organoids, which are basically small piece of tissue that we can grow and expand in a dish in vitro. And that allows us to do molecular analysis that we can't do on patients or other models. And that's really this combination of patient data and organoids model in vitro that allows us really to study mechanism of disease and to hopefully identify new treatment.
The really interesting thing about the organoids is that, as I understand it, they have 3D properties as opposed to like the kind of research and the kind of cell propagation that has been done in lab dishes maybe in even a few years back. What are the advantages of these organoids and the properties they bring?
The advantages of those systems is that you really have a representation of the cells of the organ. Okay, so you take cells directly from the liver of a patient, put that in a dish and grow it. And they conserve their characteristic. in terms of identity, so if it's liver cells, it's liver cells, but also in terms of disease. So no, if there were disease in the patient, there are also disease in the dish. That's really allow you to have a picture, basically an instantaneous picture of the tissue of the patient. And that's really unique because all the other systems we have are either based on cells that are transformed, so tumor cells that are not natural cell type at the end, or cells that are generated from human proven stem cells that are not fully functional, or at least are not such a precise picture of the tissue of the patient.
So you essentially retain the actual qualities of a liver cell or a diseased liver cell, so they don't change or turn into a more general type of cell. They stay the same, so you can actually repeat the experiments.
They stay very similar. That's the most similar model we can have from in vivo. Of course, they're not identical because they are not the tissue itself from the patient. There's nothing that will ever be a liver that is in a human being that we can grow in a dish now, simply because the complexity of the human body, the interconnection with all the other organs is impossible to fully reproduce in a dish. But they are the best model we currently have to really have an access to the biology of the organ and the tissue.
If I were to look through a microscope, what would I see if I look at one of your organoids? I probably have this very wrong concept of like a tiny lump of meat in a dish. What do they look like? Is it possible to describe it a little bit to us?
Yeah, of course. So they look like they are sphere. They grow as what we call spheroid. Of course, they're extremely small. You need a microscope to look at them, but they look like a football. And they are empty in the middle. They have a lumen, and they have a layer of cells, and they're very round. And they grow with different size, but basically they have all the same morphology.
And what does it take to create an organoid like that? You use human cell material as a base. And what else does it take? What are the kind of conditions that you need in the lab or in the dish? Do you need a particular type of source material?
So, of course, we need patient material, no tissue from the patient, which are the start. And the way we do so we then cut this piece of tissue in small pieces that we grow in a 3D, in what we call a hydrogel, which is basically some sort of gel. It's like a jelly, where we have the cells that can grow like that in 3D. We feed them with medium that contain different growth factors, nutrients and so on, so that they're happy, they can grow and so on. And we then, to expand them, what we do is that we collect those organoids, those spheroids. We break them in small pieces, and every small pieces we're going to reform a big spheroid, and that's all we basically grow them. And we can grow them for 15, 20 months with a 2-month problem.
The goal, I imagine, is not necessarily just to grow them and grow them successfully, but also to be able to manipulate them and try things out with them. Because at the end, the goal is to be able to use this kind of research and the results of it for therapy. So do you actually manipulate the cells that you've created in a way to simulate diseases or use drugs to try and see how they react?
So yeah, we do a lot of different things. We, for example, feed the cells with fat so that they become fat, like in a fatty liver. And you can see that they have small lipid vesicles inside them, and so they become very full of this fat vesicle. We can, of course, put some drugs on them. We did a lot of work on COVID-19 at some stage also, because those organoids can be very easily affected with the SARS-CoV-2 virus from COVID-19. And we have now developed drug against SARS-CoV-2 using those cells. We can do genome editing, so we can do genetic modification on those cells. And for example, what we're trying to do now is to induce mutation in healthy cells, so cells from a donor that is healthy, and we're inducing mutation to see how if we can basically induce cancer in those cells by reproducing mutation that are supposedly known to induce liver cancer. That allows us to determine the function of all those mutations in the process of tumorigenesis. And so we're doing a genetic screen, we're doing a lot of different things on those cells. In fact, you can do almost so many things as long as you're in vitro. It's an amazing source of experimental manipulation, for sure.
What are the most exciting directions your research is taking, or the most promising ones? Or is it all important as a groundwork for future research?
So, no, it's very difficult to say because all the projects that we do in the lab are exciting, otherwise we would not be doing them. So it's very difficult to give you a favorite one. I think what is very exciting for us, is that, as I told you, the liver field for a long time was a bit behind in terms of progress, and especially in terms of knowledge and so on. That's changing very fast. So we are in a field that is booming now with a new discovery every weeks or months, with new knowledge, with new model system, with new technology and so on. And that's super exciting because we are really the thing that we are at the forefront of this new wave of discovery. And it's very exciting to see that we can really have an impact, and all the projects we develop are part of this massive progress. And so all the work we do on liver regeneration, on developmental biology, on cell-based therapy, it's for us it's super exciting because it's really topical right now. I think we are really lucky because we're at the right time, right place to do this kind of work.
It's not possible, probably for many years, to stick one of your organoids or like a bigger version of it into somebody and a patient and expect it to fix their disease. What kind of time frame are we talking about? Is it's like if you're a gardener, you won't be able to see a tree that you plant now in its finished state like 100 years later. How confident are you that you will see the results of your work being actually used in therapies to treat patients?
We don't need to go for cell-based therapy to see application. The model we have developed in the past are really used now for drug screening in pharmaceutical companies and other now to validate drug, to validate target for drug development and so on, and to validate new therapy. So we already can see some level of application of the work we've been doing. Of course, now the direct impact on patient is a key objective, and that's really something we like to achieve. It's always difficult to give you a timeline because we don't control the timeline. Now, I think we need to be very clear on that. We're not the people that decide when those cells will go into patient. There's a lot of aspects that are not directly controlled by us. We already just know we need money, we need funding, we need all these kind of things to be able to do that. And that now takes time, that takes a lot of resource. And we're not the one that decide to invest in our lab. That means that there's a whole process that need to be achieved. We have several programs, and some of them are already progressing toward the clinic. And the hope now is that at least for some of those projects, we could 5-10 years after the first clinical trial. It's important to see that there's a clear, the goal here is to have a clear balance between risk and benefit. And that also will determine which disease, which patient, which type of therapy will be applying first. That's really a key goal, and we are working very hard towards this objective.
How patient do you need to be as a scientist to do that kind of work and know that it's probably going to take a long time or also more research possibly done by other people to actually come into effect at some point?
I mean, that's a very important question. Our drive as basic scientists is now, of course, to see the work that we are doing into patients, but our day-to-day satisfaction is based on the data we generate in the lab and the knowledge we generate from our data. And the new technology, the new understanding of basic mechanism, and so on. So, of course, we are super excited, but we're not only on the clinical development, because otherwise, it would be very frustrating and we'll become very desperate. So, it's a key objective for us. That's really the final objective of our work. But our satisfaction in the lab now is the day-to-day work in the lab, the new discovery, the new technology, the data we generate, the new understanding is really our primary objective.
Usually the non-scientist public only hear about the successes, of course, the big leaps and innovations in the research field. But surely there must be a lot of setbacks as well, or failed attempts to even get to a stage where you can see research results being made use of in a practical way or in a way that furthers the research. How do you deal with that kind of discrepancy?
Well, first, it's true that the general public will hear only 0.001% of what is discovered in labs because a lot of things that are found in labs are never reach the general public. Resilience is a key part of our job. We are researcher, so that means that we research. We don't find every day. That's clear. So that's really part of our training and of course one of our qualities that we need to be capable to accept that things won't work, that we won't find what we want, and that we need to spend a lot of time, energy in sometimes just redoing things for them to work better. And that's really part of our job. And I think I don't know any researcher that would not be very good at managing this frustration.
Apart from having to be patient and being able to manage frustration situations, what would you say is the biggest challenge you face in your work?
I would say I would like really to have more time to focus on science. That's something that I know. That's for me personally. That's one of the limitations. Is that I have a lot of administrative constraints and other that really block me to spend more time at reading papers, discussing with my team, doing research directly. And that's probably one of the major limitations when you become a bit more senior, as I am, that you just have less time to think and to reflect on science.
If you go into work like every day, do you have like a vision of what you want to end up achieving? Do you have a scientific result in mind? What keeps you going ultimately?
What is, I say, it's discovery. It's seeing a new result in the lab, even a tiny thing, it's a new graph, a new staining, a new mechanism. That's what really drives us. Well, I'm also really happy and excited when we can get to publish our data and that we can show to the field that we are making progress and share the knowledge and demonstrate that we have generated knowledge. Working with my team also, that's something that's really, able to have the intellectual challenge and intellectual interaction with my group is really something that I really enjoy. Promoting the new generation also, helping them to achieve their objective and their career objective. That's something that is also something that is strong motivation for me. And at the end, is doing research. That's the key, and with all the different aspects that it entails.
I'm sure you're not going to run out of either ideas or material to do more research on. It's a very positive outlook there coming from Ludovic Vallier. He has been our guest on this episode of AskDifferent, the podcast by the Einstein Foundation. He and his team are, among other things, doing research on liver organoids, hoping to sometime in the future be able to derive treatments and possibly an alternative to organ transplantation as it is done now from this very work. Thank you so much for your time and for being with us today and giving us some insights into this very big and very exciting field of research. Thank you so much.
Thank you for the opportunity.
You've been listening to Ask Different, the podcast by the Einstein Foundation, where we speak to scientists and researchers in inspiring and exciting fields about their work and their findings. And you'll find us wherever you get your podcasts from. Do subscribe if you haven't already. My name is Doris Helpold. Once again, thank you for listening. And until next time.
Ask Different, the podcast by the Einstein Foundation.